Hello, everyone, and welcome. Thank you for joining us today for the sixth and final webinar in our current series titled The New World Screwworm, Past Lessons, Present Programs, Future Frontiers. My name is Justin Overcash, and I'm with the Foundation for the National Institutes of Health, where I work as part of the Gene Convene Global Collaborative. Gene Convene's mission is to support and advance best practices and informed decision-making for the development and governance of genetic and biological control technologies to improve public and animal health. We do this by convening diverse expertise, identifying key scientific and policy questions, and supporting transparent information sharing and capacity building across sectors. This webinar series focuses on the New World screwworm, a pest that historically has caused devastating impacts on livestock, wildlife, and human health, and whose successful control stands as one of the most significant achievements in area-wide pest management. At the same time, recent detections and northward spread underscore that this is not just a historical story, but a contemporary and forward-looking challenge. Across this series, we have examined past lessons, present programs, and future frontiers, including new scientific tools, technologies, and policy approaches that may strengthen long-term control and prevention. Before we get started, I'll cover a few housekeeping reminders. If you experience any technical issues during the webinar, please email events@fnih.org. Your video and audio will be turned off during the session. Today's presentation will run approximately 45 to 60 minutes, and please feel free to submit questions at any time using the Q&A function. We will address questions during the discussion period at the end of the presentation as time allows. This webinar is being recorded, and recordings will be posted to the Gene Convene Virtual Institute YouTube channel following the event. Today's webinar is titled Screwworm Male-Only Lines, Past, Present, and Future. We are pleased to be joined by Max Scott, professor in the Department of Entomology and Plant Pathology at North Carolina State University. Dr. Scott received his BS in biochemistry from the University of Western Australia and his PhD in molecular cell biology from Baylor College of Medicine. He went on to spend 16 years as a lecturer in genetics at Massey University in New Zealand before joining NC State University in 2010. Over the course of his career, his research has focused on the development of genetic tools for insect control, particularly through the use of transgenic approaches. At NC State, Dr. Scott's lab has worked extensively on developing genetic biocontrol strategies for several insect species, including New World screwworm, the Australian sheep blowfly, and spotted-winged Drosophila. A major focus of his work has been on development of male-only strains, which have the potential to significantly improve the efficacy of control programs based on the sterile insect technique. Today's presentation will explore the scientific foundations, current progress, and future potential of these approaches in the context of screwworm management. Before we begin, a reminder that I will be moderating the discussion following the presentation. Please feel free to submit questions in the Zoom Q&A chat at any time. With that, I'll turn it over to Dr. Scott.
Okay, so we're going to talk about screwworm male-only lines and so what we did in the past to get here and where I think we could go. All right, so just a reminder, screwworm is New World screwworm's a devastating obligate parasite, major pest of livestock, and that's why we're having this session. The sterile insect technique was developed by the USDA, led by Nippling and Bushland. And the central idea is that insects are sterilized by radiation, and if a female out in the field mates with a sterile male, the offspring, the embryos will die due to massive sort of aneuploidy. All right, so for suppression, typically the program has used about a 10-fold excess of sterile males and females. They're releasing both sexes, and suppression requires the sterilized male to mate with a fertile female. And you have to do repeated releases to get eradication. But it is a species-specific suppression, so it's considered a green technology. Obviously, it worked, right? So we've had this before, but this is, just remind you, starting in Florida in the 1950s, progressing down to Panama in 2002, 2006, screwworm was eradicated. Obviously, now it's coming back, but it was very successful, and the border was maintained for a long time. But it was expensive. I think the eradication program probably cost about $1 billion. So the central question I was interested in, really from a postdoc I did on Mediterranean fruit fly, was, how can we improve the efficiency of SIT to make it more affordable for other species? And we knew about this. One way to do this from right way back at the beginning, um, well before he published, Nipling corresponded with some of the prominent geneticists at the time about his idea for using sterilized screwworm. So here is a copy of a letter he had written earlier to Hermann Muller, and we know that Muller replied like 2 or 3 days later and outlined why I thought this was a great idea. But one part of his letter I thought I would show here was Muller says, of course, it's going to be a tremendous amount of work to separate males from females, but then he goes on to say why that would be a great idea. For the efficiency of the treated males as competitors of the other males would be reduced in this way, since they would only engage in a fraction of the matings with the wild females if they had the artificially bred ones with them. So releasing only males should improve the efficiency of SIT, and we know that's true for the Mediterranean fruit fly. Still remains to be shown for screwworm, but reason to believe that would be the case. So then this is where I'm a do genetic engineering, where biotech could come in. I thought, biotech enhanced SIT, release only sterile males. We should get more efficient genetic suppression because the released males will only mate with wild females in the targeted region. We should save diet costs if females die at the embryo or first instar stage. It's the larval diet that's really expensive in the screwworm program. And particularly for screwworm, it's not necessarily true for all insects, but the dose of radiation required to sterilize males is quite a bit less than required to sterilize females. So by using a lower radiation dose, that should improve fitness of the released males. And that's something that is being looked at right now in Panama. Okay, so in putting this talk together, and I realized I actually hadn't given this talk before, to make sense of what we did at NC State, I need to briefly explain what we did prior to that in New Zealand. So as Justin mentioned, I was a lecturer in genetics at Massey University in New Zealand for 16 years, from 1993 to 2009, before moving to Raleigh. And in New Zealand, we were sort of thinking about this. How do we make a male-only strain? So one way to do this is to activate a lethal gene only in females. My background was on Drosophila sex determination and dosage compensation. So another way I thought about doing this was to transform XX flies to males. I'll come back to that later. Whatever we do, we need a conditional system for controlling gene expression because we need to be able to mass rear the strain and only in the release generation throw a switch and produce only males. We initially tried to use temperature, couldn't get a temperature-based system working. So then we switched to the tetracycline system, which had been developed in the early 90s mostly for mammalian systems, but it worked well in insects. And in this system, the transcription factor is TTA. It's a fusion of the DNA binding domain from the TET repressor from E. coli with an activation domain that worked in eukaryotes. This TET repressor DNA binding domain is incredibly specific for a TET operator sequence from E. coli. And this sequence doesn't occur naturally in insect genomes. So we're only really going to regulate the gene that we're targeting. But the real beauty of the system is that this binding is strongly inhibited by the presence of tetracycline or a derivative like doxycycline in the diet. The antibiotic binds to the DNA binding domain, changes the conformation, and there's a major change in the binding affinity for DNA. So it's a really beautiful off switch. And then the system we put together, so the lab in New Zealand was pretty small, but I did manage to get funding from farmers, from the sheep farmers in New Zealand through Wolves of New Zealand. And I recruited my friend Jörg Heinrich, who I had known in Greece, to move to New Zealand. And so we worked on this together. So this is the system. So central is TTA, the transcription factor that's inhibited by tetracycline. We only wanted females to die, so we used transcription. We used a female-specific promoter from a yolk protein gene. So only females are going to make TTA, and then we want to kill them. And we considered a number of effectors, but we settled on this gene called HID, which doesn't have a very impressive name, unlike other pro-cell death genes like Reaper and Grim, Sickle. But this is actually probably the most effective at killing the whole organism. So we're going to induce massive amounts of cell death, and we put this gene under the control of TTA by having multiple copies of the binding site, the TET operator, and you need a core promoter, a minimal promoter to assemble the basic transcription machinery. And so this worked, produced only males and looked like a good system. So New Zealand didn't have a lot of pests, insect pests, and the one we that was there that we got funding to work on was the Australian sheep blowfly, Licilia caprina. It had been a pest in Australia for many years. You know, my grandfather was a sheep farmer and was always dealing with fly strike. Um, but it didn't arrive in New Zealand until 1988, and it's a pest because the females look for typically a wound or a scratch in the uh, in the sheep and lay their eggs in the wound, and the larvae that develop, consume the flesh and cause meiosis. So this would sound very similar to screwworm, but it's not considered an obligate parasite. It's considered a facultative parasite. Okay, so that was the pest. To do any genetic engineering, we had to develop methods to make them transgenic, and this took us a long time to figure this out. Um, we tried a different number of different vectors. In the end, piggyback is the one that worked. And we tried a number of marker genes, and the one that worked initially was, we got this from Al Handler, it's just EGFP driven by a Drosophila promoter. It wasn't that active in Licilia, it behaved more like an enhancer trap. But we did pick up some transgenic lines, and we published this. But this was, I think we felt like we were missing some transgenic lines because the marker was so weak. So we re-engineered the marker using a promoter from a Licilia HSP83 gene. This is a really strong promoter, driving a brighter fluorescent protein, ZS green. Carolina Concha led this work. We also made some other changes. Esther worked on this in terms of how to better handle the embryos, better needles, and then we achieved much higher transformation rates, typically around 10%, although it varies a lot from experiment to experiment, and the flies were easy to identify. So with a transgenesis method, then we turned our attention to assembling this female lethal system. So we isolated a promoter from a Licilia yolk protein gene, hooked it up to a laxid reporter, made transgenic flies, and there was good news and bad news. Good news was, yes, the promoter was female specific. So we could use it to get female expression of TTA. But the problem was the expression was only activated in adult females and only after they'd been fed a protein meal. And so this is far too late to be useful for building a sexing strain. So I mentioned my background was in Drosophila, X chromosome dosage compensation, and sex determination. And so in parallel, I was interested in figuring out the sex determination pathway in the sheep blowfly. And it's a bit different than Drosophila. The bottom is the same. The double sex and fruitless are sex specific as in Drosophila, and they're controlled by transformer combining with transformer 2. The difference is that transformer is autoregulated as in the Mediterranean fruit fly and the housefly, and it's repressed by a Y-linked male determining gene in males. We still need to identify that. Well, I say it's autoregulated, so this was work for Carolina Concha. She isolated the transformer gene, which was not trivial, and found that females produced a shorter RNA that had a long open reading frame, much longer than Drosophila. But males had additional exons with in-frame stop codons, so they produced shorter, probably non-functional peptides. And why we think this is autoregulatory is because the sex-specific intron had multiple binding sites for the TRA-TRA2 complex. And the sequence was pretty much the same as found in the double sex and fruitless genes. So it's highly likely splicing is autoregulated. So we had a different means now for getting sex specificity. This system is activated very early in the embryo, as would be expected. Females inherit the female form from the mother. This is what the lower band is showing. And unless something happens, all the embryos are going to develop as females. So what happens is pretty early on, 2 hours is around when cellular blastoderm forms. The male determining gene on that Y chromosome is activated, and somehow inhibits the female mode of splicing, and you start seeing the male form appear. All right, so that's the work we did in New Zealand, and what really helped in getting us going at NC State was I didn't move alone. So my wife, Esther, moved with me, which was great. Esther's sort of biochemist by training, but she had become an expert in handling Drosophila and Lucilia and doing the transgenesis. Fang Li had been in the lab for several years, and she agreed to move with us to North Carolina, and she's an expert molecular biologist, really good. And not initially, but I think after we'd been here a year or two, Carolina rejoined the group, and she led the work in Panama. So we had well-established teams to get us going at NC State. Right. At that time, we were not allowed to work with New World Screwworm. That was a quarantine pest. It might be changing now, but we had to work on something else. So we knew how to work on Lucilia caprina. We had worked that out in New Zealand. So the idea was we would work on Lucilia, develop and evaluate systems there in that species. And if they look good, then transfer them to Panama to be tested in the New World Screwworm. And these are both blowflies. Okay, one of the first things Fung did was isolate the transformer gene from primary screwworm and also secondary screwworm, Cochlearia macellaria, and another Lucilia species. And they all have the same sort of organization, a sex-specific intron, multiple TRA-TRA2 binding sites, and male-specific exons with in-frame stop codons. So we had the sex-specific intron from screwworm transformer to use in constructs. And we really felt like we needed to have success pretty quickly. I moved here as an associate professor without tenure. And so the quickest thing we could build was a single component system. These had already been shown to work well in a number of insects by Luke Alfie and colleagues in the UK. And so TTA again is central because it can be switched off by tetracycline. In this auto-regulatory system, many copies of the TET operator put upstream of just a core promoter. So a little bit of TTA is going to feed back and make more TTA. And eventually females die and probably die to a general interference in transcription. So the great thing, again, it's switched off by tetracycline. And to make it sex-specific, we used the intron from transformer, because by this point we had the primary screwworm transformer intron. So we tested it in Lucilia and also Drosophila at NC State, actually worked in both. And so then we were confident this should work in Screwworm, and we sent it to Panama. So this is Fung and Esther's data, Lucilia caprina line. Then most of the lines showed the same behavior, that with tetracycline, males and females survived. Without tetracycline, we only got males. So it looked pretty promising. And Carolina put this into New World Screwworm, and she made many lines. I mean, really, she made a lot of lines. I think we've still kept a couple of them, and they worked really well. Produced only males, and when assessed for most of the characteristics that would be important for production, they look comparable to the production strain that was used at that time, Jamaica 6. I should say one of the benefits of working in Panama at the USDA research lab is Carolina was really well supported by the technical team there, led by Augustine, with really great support from Mario and Gladys, and I think Trinidad at the time. So when I say Carolina, keep in mind that there was a great technical support team working in Panama. And we actually went on and did a field test with radiation sterilized males from one of these lines in 2018. It took us 3 years to get regulatory approval to do that test. It was never published. COPEC has that data. And the line probably was not comparable, didn't quite disperse as well as the Jamaica 6. But we had problems trapping the males. We'll come back to you later. Okay, so these strains look pretty good in my opinion, but they did have a disadvantage, is that the females didn't die till late, pretty late larvae pupal stages. And so before they die, the females are consuming a lot of diet, which is an expense for the program. So we were asked to develop female embryo lethal strains. And so again, we turn to the back to what we did back in the beginning. New Zealand, we used the HID gene, this time from Lacilia, used the screwworm intron, so it's only female specific, put it under the control of TTA, and because we want embryos to die, we used a promoter from a Lacilia cellularization gene, which was active in the embryo, right? So only in embryos, you'll get TTA made, switch on production of HID, and females should die due to apoptosis. And we knew this system should work based on work that had been done in tephridids by Ernst Wimmer and Mark Schetteling and colleagues. So by this point, Ying Yan had joined the lab, first as a visiting grad student from China and then as a postdoc. And he made a number of sexing strains, and they all look good, right? The females died in the absence of tetracycline, and lethality was very early, embryos L1. So we sent this to Panama, and Carolina and the team there made a number of sexing strains, and at some point Ying went down and joined in this. So we had a number of two-component strains, right, with a driver and effector combined. So one was marked with red and one with green. So we had to have two markers. And they all behaved well, tetracycline kept females alive. Without tetracycline, the females all died. We also had one we called an all-in-one where the driver and effector were in one construct. And I'll come back to that later. So these looked pretty good in a number of characteristics for production, but there were a couple of problems. One was that although we selected this promoter, this bottleneck promoter, because the gene was really just expressed in embryos, the driver had some activity in ovaries. So why was that problematic? It's problematic because for these embryo lethal systems to work, we cannot feed tetracycline to the adults, because otherwise the females pass the tetracycline on to their offspring they'll load up the eggs with tetracycline, and that will switch off the embryo female killing system. So we need to not feed tetracycline to the adults. And when we did that for a number of these DR2 lines, what we found was that the females were sterile. And it turned out because they were making some TTA in the ovaries, which was activating HID and inducing cell death in the ovaries. The workaround that Ying found was he could get around the sterility by feeding females a low dose of tetracycline for a few days after emergence. And we did this first in Lacilia and then also in Screwworm. But the feeling was that this was a bit too fiddly to be useful for mass rearing. So we needed better TTA drivers. So Ying initiated this work and was picked up by a former grad student, Megan Williamson. So we made new drivers using a nullo promoter from Lacilia and a promoter from a related gene, or no, not related, a linked gene, CG14427, which I think has a new name now. That was from Cochlear Meyer. So these are the new drivers, DR6 and 7, that based on the RNA-seq data we thought would be more embryo-specific. And the reporter is a DS-red gene activated by TTA. So wherever TTA is made, we should get red fluorescence. And what Megan found is that indeed, yes, the DR2 and a DR3 driver I didn't talk about showed red fluorescence in ovaries, particularly the immature stages. The driver, without the driver, what we see is green fluorescence from the marker. And these new drivers really did not give us any expression in the ovaries. So we thought this looked pretty good. And Ying was able, and Megan were able to put together sexing strains that with tetracycline, you got males and females. Without tetracycline, females died, and they appear to die quite early in development. So we thought we had good sexing strains. And then we sent it to Panama. At this point, Alex Arp had joined the group initially as a postdoc, and then he was hired by ARS. And in this case, the system didn't quite behave as it did in Lacilia. And so we probably, in hindsight, kept our strategy of testing in Lacilia and then trying in New York Screwworm a bit too long. We probably should have developed the secondary Screwworm as a model faster than we did. So what happened with DR6? Yes, he did make sexing strains, but for some reason, even though this looked like a strong embryo driver in Lecilia, in Screwworm it was not. And the females didn't die until quite late in development as pupae. But he did get, only males produced without doxycycline. The DR7 driver was embryo specific and could make very effective sexing strains. But this promoter was really strong in New World Screwworm, and the DR7 lines were difficult to handle. But at least we're making only males, and the lethality appeared to be very early in development. So a problem with these DR7 males was in the behavior. This also was true for DR2. So we do two kinds of mating behaviors. We do a mating success where the females are placed with a limiting number of males, just one type of male, and we ask how many females mate in that time. And so we call this mating success. And for the DR7 males, so the mating success was comparable to the parental strain Jamaica 6. So by that behavior assay, these strains look good. But when we did a competition experiment, sort of one Jamaica 6 male, one Jamaica 6 female, and one transgenic male, all the strains were outcompeted by Jamaica 6. So we seem to have a mating competitiveness problem. We thought, trying to think why this might be. One thing that occurred to us is, if we're getting some leaky expression of HID in the males, this could be reducing their competitiveness. And so we thought maybe if we make the system more sex specific, we can get around this issue. And this was the work of Adamalia Vargas Lohman. She's a professor at the University of Panama who took a leave from her position to work on Screwworm for a couple of years, which we're glad she did. And so to make the system more sex specific, we added the tri-intron to the TTA gene. So now only females should make TTA. We also changed the promoter because in the initial Screwworm genome project that I led, we identified the halo gene as being highly embryo specific. And there were two link genes close to it that were related that also looked very embryo specific. So we thought this might be better promoters. So these new drivers were DR9 and 10, combined them with the existing effectors, which were already female specific from the tri-intron. And this work was recently published. So Adamalia put together several sexing strains. She called them 553, 554, 561. And they produced males on diet without doxycycline. It looked pretty good. And two of them, the females were dying early in development, embryo L1. One of them, the lethality wasn't until late pupal stage. With the mating success assay, much as we'd seen before, they are no different than the parental strain, Jamaica 6. So this looked good. Question was, were we doing any better in competition? And the two embryo strains were outcompeted, perhaps not as greatly in this one, but they were statistically significant. The one strain which was competitive was the pupal lethal strain. So didn't have an ideal strain that was embryo lethal and competitive. So to summarize where we were on all this male-only work, we had several embryo and pupal female lethal two-component strains, produced only males on diet without tetracycline. And, most of them, the males showed high mating success, right? So there's no mate choice. The females, mate quite well with them. But all the female embryo lethal lines, the males were outcompeted by wild type in these small cage tests we were running. So we concluded after all this work, which was a lot of work, the new lines made with the DR6, 7, DR9, DR10 drivers were not an improvement over some of the original DR2 sexing strains, particularly an all-in-one line called TD1. So while many of the DR2 sexing strains had this problem of female sterility, this line never did. All right, so gone through this fairly quickly, but it gives me a bit of time to talk about future. So in the immediate future is this, it's an old line, but it's the one that's being currently worked on. So Ying made an all-in-one construct. So in the one construct, we have EF1, and then we have the DR2 driver and a DS red marker. So this makes it a little easier to handle because it's just one construct. With two separate constructs, it's a little difficult to sort of make them double homozygous. And this TD1 strain, we've done a lot of work with it over the years. And the biological yield, the things that are important for mass production, it seems comparable to the parental strain. It's also comparable with longevity and mating success. Right, so on all those measures, it's equal to production strains. It was outcompeted in the small cage tests. Whether this reflects what will happen in the field, we'll wait and see. But anyways, this is the line that Alex has chosen to go ahead with. It's been crossed into the pan-CR background. So the mass production strain has been changed from Jamaica 6, where all the work was done in Jamaica 6, because it appears that the Jamaica 6 strain was not succeeding that well in the field. And so the new strain was developed from larvae collected in Panama and Costa Rica, so you get this pan-CR name. And so this strain now in this background is going through EPA registration. It's called NovoFly, and it's available for public comment. And I think I've put the link to that comment in the chat. So that's the one that's going forward. And of course, we just had the Screwworm Grand Challenge, so I put my 2 cents worth in. I think we could actually, there's still room for improvement potentially in the two-component system. One avenue we've been thinking about is changing the promoter. So we've done RNA sequencing on single embryos, and it seems that the final adult form of the male splice vitrea and the female splice vitrea doesn't become really well set until several hours into development. And so maybe by looking for promoters that were active, really early, like 1 to 2 hours, we might have been expressing TTA before the splicing signals were completely fixed. So I'm thinking that maybe it might be good to look for late embryo or even L1 promoters to drive TTA to achieve early lethality. And the other idea comes from our work. So in parallel to all this, we've been working on Drosophila Suzuki. And, HID is quite a difficult gene to work with as an effector in Suzuki. Even with, really weak core promoters, just leaky expression of HID is difficult to work with. So we've been looking for alternative effectors, genes that when expressed cause lethality. And we've identified some that we think could be considered for this system that may have better fitness parameters. So who knows? I should say my funding to work on these systems ended several years ago. Down in Panama, we've become concerned about this mating competitiveness assay, just how realistic it is for telling us whether something will succeed in the field. So Alex has been working on this. And so the old assays were done in small lab cages like this. And Alex has now developed a new system, a much bigger cage with sort of artificial trees, resting points where the males can sit. Because we think this is what happens in the field, that the males are sitting on branches and grabbing females as they fly by. And he's put the small cage in here so you can see for a comparison. It's not that we run the assay in the small cage within a big cage, but that's what we do. So I think that's promising. And Alex was telling me that using these cages, the TD1 NovaFly is performing quite well when exposed to lower doses of radiation. Right, so then going forward, how to make a male-only strain. So I'm still interested in this system transforming XX flies to males. So the advantage of doing this is you're not losing any of the eggs. All the eggs produced will go on to produce males rather than killing half of them. And we have, I know I'm talking about the future, but we have worked on this a bit in the past. So Megan and Ying worked on this. So the idea was use our existing embryo drivers, but have them hooked up to an effector that produces, and it's an inverted repeat of an exon from transformer. And so when it's transcribed, the RNA is going to very quickly fold back into a long stem double-stranded RNA. It's going to be processed by the RNA machinery to produce, short interfering RNAs that would um, knock down RNA. And potentially, if the knockdown is sufficient enough, the XX female should develop as males. So this work was published a few years ago. One of the things about the TET system is it seems quite sensitive to position in the genome, and we have no control of position with piggyback. So we get a number of lines, and we have to test many lines to find ones that work. And Megan saw a range of phenotype. Some lines didn't work at all, we just got XX females. Some worked a bit. The female ovipositor showed this bent phenotype, so that was sterile. And then some were somewhat masculinized, had some masculine tissue, or they were externally looked like males, but internally not. Occasionally, we saw a fully transformed XX fly, but most of the time, the XX flies died, and they died at the pupal stage. And Megan went on to show that this was due to a problem in X chromosome transcription, X chromosome dosage compensation. So there's not many genes on the X in Lucilia or Screwworm, probably no more than 100. And they are dosage compensated, so they're equally expressed in males and females. And it appears if that dosage compensation is not regulated correctly, it's lethal, as in Drosophila. So we think transformer controls dosage compensation. And to get this, we had to actually knock out expression in the mother, in the ovaries, in addition to the embryo. So it didn't look promising, and also Alex tried the RNAi approach in Screwworm, and the results were not good. We had a lot of off-target problems. So more recently, Alexis, who just graduated from the lab a couple of months ago, a main part of her thesis was, returning to this transformation approach, but instead of using RNAi, using CRISPR to either knock out or knock down transformer expression. So the effector in this case is Cas9 or dCas9 activated by TTA. So Cas9 will cut the gene, dCas9 will bind to it and inhibit expression. Within the construct is a single promoter from a Lucilia U6 gene driving a cassette of 3 guide RNAs. When with tRNA processing works, we should get these guide RNAs released. The idea is these guide RNAs would target transformer. And she ran a control with guide RNA targeting DS-RED that we knew worked really well. This paper has been accepted in CRISPR journal. I'm not sure it's online yet. We've had the proofs, but should be out soon. It's also available on bioarchive. Okay, so I'll just summarize what she got. So TTA worked well. We got high levels of expression of Cas9 and dCas9. But with a major surprise was high levels of dCas9 were not good for the fly. They were poorly tolerated. We had a lot of issues with lethality. With the control gene, DS-RED, Cas9 was very effective at knocking it out just with a single guide RNA, but dCas9 was not. So dCas9 does not look like a good approach. But with trial, we had a problem, and we didn't see sex transformation, and it appears to be a problem with guide RNA production. That tRNA processing step, I think, is not working well. Alexis did see sex transformation when she supplied additional guide RNA by microinjecting embryos. So I'll just show you a little bit about that data. So this is the knockdown of the DS-RED by inducible expression of Cas9 with tetracycline. DS-RED was bright, and without tetracycline, the fluorescence was notably reduced. And we collaborated with a group here to show that using mass spec that the protein level was knocked down substantially. But with dCas9, we did not see any change in fluorescence when we removed tetracycline. And then the sex transformation data, how clear this is with a control female, control male. And she injected a mix of guide RNAs into embryos from these two-component strains. So she had strains that were homozygous for either DR3 or DR7 driver, and one of these Cas9 effectors that should express three guide RNAs for TRA. And she called these TRA246 in this example. They're cutting at three different positions in the gene. They're quite far apart. So if there's no cutting, you can't actually detect the band with the PCR conditions that she used. But if there's any cutting, we should get deletion products. And in the guide RNA injected embryos, in addition to seeing partial sex transformation, she saw evidence for the deletion products being produced. But in the two-component strain that, if everything worked well, should be producing trial guide RNAs, but, saw no evidence for cleavage. So I think this is a promising approach for producing only males, getting back to the theme of this talk, but it's going to require some modification. We probably, I think, should use a separate promoter for expression of each guide RNA and forget about using dCas9. Right, so a related technology, I think, that could be used to produce only males is called PGSIT for precision guided SIT. This was developed by the Omar Akbari and colleagues at UC San Diego and is now commercialized by Agrigene. And so I think this is quite a promising technology that could be developed in Screwworm. So to do this, you have a Cas9 female. So rather than an inducible system, this just makes Cas9. The promoter that's been used in Drosophila is from the Vasa gene, which has activity in the germline and the soma. So a Cas9 female is crossed with a guide RNA male. The guide RNAs would target, in the case of Screwworm, we'd have to target transformer. So we'd transform XX females into males and target a gene that's required for male fertility. The one used in Drosophila, beta-2 tubulin, should work in Screwworm as well, and the gene is there. We've seen it in the genome. So when you do this cross, the Cas9 guide RNA complex is assembled in the embryos, and the females should develop as males, and the males should be all sterile because of the activity of knocking out beta-2 tubulin. I think it's worth doing. There are a couple of challenges with this, apart from the engineering, right? Engineering, so you get Cas9 expressed where you want and guide RNA where you want. The main challenge is you have to do this cross at scale. So you need a means of sex sorting so you can obtain virgin females from the Cas9 line and males from the guide RNA line. And I think there are ways of doing this. I don't think it's a challenge, but I think it's doable. So I think this could be a feasible approach going forward for producing males. And of course, no need for radiation because the males are sterilized by CRISPR. So in the last few minutes, just talk about the conditional system for controlling gene expression. So the tetracycline off system, great system. Really you can fine tune it with that tetracycline. But tetracycline is an antibiotic, and it does change the microbiome, both in Lucilia and in primary screwworm. And we did some work on this. Alexis showed that in Lucilia, the microbiome's changed, and there is a lingering effect into the next generation, and it does reduce male fitness a bit, but was significant. In primary screwworm, Alex was doing experiments in parallel, and yes, tetracycline changed the New World screwworm microbiome, but didn't seem to impact fitness. So some species difference there. Nevertheless, because tetracycline is an antibiotic, there's interest in other ways of doing this. And there are other small molecule-based systems. The quinic acid or Q system is used extensively in Drosophila and could be potentially tried. Other small molecules we've worked on are auxin and vanilla. We've done some work in Drosophila that's unpublished. So I think, there's potential there of different small molecules. And it may be worth, coming back to temperature, which is how we started this way back when we first started in New Zealand, trying to get a temperature-controlled switch. Because this is how the classical genetic sexing system works in Mediterranean fruit fly. And, my postdoc, one of the postdocs I did was on medfly at the IMBB in Crete. So the classical medfly genetic sexing system works by having the white pupae gene and the TSL, the wild type TSL gene translocated to the Y chromosome. So it's a chunk of chromosome 5 is translocated to the Y. The autosomes remain homozygous for the white pupae mutation and the TSL mutation. And so as a consequence, because the normal genes on the Y, male pupae are brown, female pupae are white, and those females are very sensitive to increasing temperature and are killed by exposing the embryos to high temperatures in a water bath. The males survive. So it works really well at scale with medfly, and there's been a lot of interest in adapting this system to other flies, mostly other tephridids. Potentially, could this system be ported to primary screwworm? Well, I mean, we now have pretty good, I think, Y chromosome assembly. Paul Heckner spoke about that a while ago. And Danny Paulo and Alex and Megan Williamson in my lab, developed gene editing protocols. It's pretty, works pretty routinely. So I think potentially we could do the editing. The challenge is more on my mind is, can we even use temperature, right? So New World screwworm embryos and larvae are reared, somewhere around 37 to 40 degrees Celsius. And my conversations with Copeg is they're not all that keen on changing the temperature used for rearing embryos. The New World so the white pupae gene, there's an awful lot present in the New World screwworm genome, but it remains to be shown that if we knock it out, it'll have the same phenotype, that we'll get the white pupae. Hasn't turned up spontaneously in the mass rearing. The TSL gene is present in the New World screwworm. It's present in a lot of insects, but there's no guarantee that we make that same single amino acid change that the screwworm will become temperature sensitive. So there's some attraction, but I think some challenges with this approach. And the last thing I want to finish with is maybe we can forget about using radiation at all and release fertile males, right? And this approach is called FS riddle. Luke Alfie coined this term some years ago, female-specific release of insects carrying a dominant lethal. So we avoid radiation altogether, and it works because the female killing gene is inherited. And this was apparent to us early on, but I should have done some modeling. So the release males that are homozygous for the female lethal system, everything inherits one copy, the females die. The males that are produced, now they're free to mate again with the wild type females around them. So they only have one copy of the female killing gene. But, half of their offspring will inherit this, female offspring will inherit this gene and will die. And some males are produced again. So you still need to do repeated releases, but the modeling is clear that this should be more efficient than SIT, particularly considering the fitness costs of radiation. What I think is particularly attractive is to build on what Tatiana spoke about a couple of weeks ago, developing a gene drive. Coupling FS riddle to gene drive would make the system even, much more effective. Right? So what we would do, we still got FS riddle, like, for example, the TD1 construct, but attached to it a homing gene drive. And so that in the germline of these males, they would be converted from heterozygotes to homozygotes. So instead of half the offspring inheriting the female killing gene, all the female offspring inherit it. And the modeling is clear that this is a particularly effective system. So if I could get the funding, this is something I would be interested in working on. I mean, there's definitely a challenge here of with all gene drive systems dealing with resistance to drive that we'd have to think through. All right, so good, finished within the hour. I've tried to acknowledge the people in the lab that did the work as I went along. This was I'm showing a picture of my lab from a few years ago. We're quite a bit smaller now, and a number of collaborators on this project. Really grateful. Early on, I had startup from NC State But the first grant I got was from the Bragg fund from the USDA, the Biotech Risk Assessment Grant. It's been a great fund for us. It's really disappointing to me that this fund has not uh, continued in the last couple of years. It really is what funded us to make those initial screwworm male-only lines. Subsequently, we were funded by agreements with APHIS and COPEG, and NSF funded a more basic blowfly project I didn't really talk about too much. All right, that's all I have to say. Thank you.
That you said quite a bit. You covered quite a bit of ground there. It's fascinating presentation. I see we already have a few questions are popping up in the chat. I just wanted to remind everyone if you have a question, go ahead and throw it in the Q&A function, and we'll get to it. But as the host, I can ask the first question if I want to. What do you have any hypotheses on why the DCAS9 was causing lethality? That was very interesting.
Yeah, really surprising to us because there's what, two amino acid differences between Cas9 and DCAS9.
Yeah.
Don't know. I mean, they're really difficult to work with those lines. There's some reports that the DCAS9, you know, stays bound to the DNA and inhibits DNA replication. So it's one of the things we talked about in the paper. But yeah, really a surprise.
Yeah, that was
And
quite interesting.
apart from being lethal, it didn't seem to do much in terms of inhibiting expression.
All right. Let's see. The first question is kind of a multi-part one, but awesome story, Max. Really enjoyable to have the historical context. The first question is, did the low competitiveness of males also occur in the presence of TAT? There's um there's a kind of a follow-up. And what about the heterozygotes? So was there a difference in competition in the heterozygotes? And then also at low competitive. So I guess it's a question about the competitiveness in the presence of TAT, or if they only had one copy of the construct,
Right.
and also
As
low
you would
competitive.
if it's FS Riddle, right? Then
Mm,
you produce males with one copy. You would have to go back and look at the data. I don't remember if we did that. I think we thought about it, but I'm not sure we did it.
okay.
We only ever did the behavior work on New World Screwworm. Um, with Lucilia, we just developed um, just really more interested in whether they killed the females, as that was not the species we're getting ready for a field trial.
And then their next question was a back crossing question. So I
Mm.
guess once you had the lines down in Panama, did you back cross to Jamaica?
Some initially, yes, before we made them homozygous, but that is, in making any of these strains homozygous, we're doing it by picking larvae that were brightly fluorescent. So we try to take as many as we can, but there's definitely some inbreeding in making any of these lines homozygous.
And I get there's another question, but let me piggyback off that one. So I know my background was in Aedes aegypti. We worked with the Liverpool strain, very domesticated. How long? And then maybe this is in one of the talks. How long have those New World screwworm lines been in Panama, and how domesticated are they, or do they back cross on a regular basis?
Yeah, good question. So the Jamaica 6 line was established in 2006. So, and it, been in culture for a number of generations. When there were some initial problems in the field, one of the great success stories of the Screwworm was that developed a mass cryopreservation technology a long time ago, well before it was established in Drosophila. And so they were able to revive the stock by uh, bringing back those embryos that had been cryopreserved early, not long after the strain was established. So that worked a couple of times, but then I think there was evidence the strain was not working well in the field, and so it was eventually switched to Panacea. Our transgenic lines, yeah, they've gone through many generations, particularly the single component lines, but even the DR2 one's been in culture for a long time.
And then this has last question, has to do with kind of increasing, I guess, the competitiveness. Did you ever consider knocking out the fluorescent markers after the line was established to test whether the fluorescent markers are responsible for an impact on competitiveness?
Yeah, good question. We definitely have thought about it. Haven't done it. But yeah, I mean, it could be, right? I mean, they're they are making a lot of this fluorescent protein. That was one because we wanted a strong marker. You can't really see it much in the adult. Um, the 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 cuticle's quite dark. Maybe a little bit of fluorescence around detectable around the mouth parts. Um, but it's something we could do. One of the advantages of the strong marker was it was something we could pick up in traps. Um, Perhaps had to squish them, but it was a way of determining that the males caught in the traps were from the released males. So we did see this as an advantage, but in terms of addressing whether that was an issue for the competitiveness, yes, it's something we thought about. We haven't done it. We could also just change the marker um, and use a different promoter. I mean, we have a lot we could use now based because we have the good genome assembly.
Right. That is kind of I mean, there is certainly an advantage to having a fluorescent marker in there when you're doing monitoring and surveillance. Which I'm going to ask a question kind of just based off that. I know you mentioned, right, in MedFly they have the cuticle protein knocked out. Um, I guess a question I have is, right, even if you were able to do something like that in New World Screwworm, does it impact the pigment of the adult flies, or is it only at the embryo?
This was the white pupae. Is that what you're
Hmm.
looking at? As far as I know, it's just the pupae color. Pupil
Just the
color
pupae.
has changed.
Okay. Because I
Uh,
guess there would always be a question about, if you change the color of a fly, how does that impact mating behavior?
hmm.
All right, let's see. The next question. Thank you for the insightful talk. For the DR lines exhibiting either strong embryonic lethality or increased mating competitiveness, to what extent do you attribute this to position effects? And have you explored the use of gypsy insulators in piggyback constructs to buffer against genomic context variability?
Good question. We have not explored that. It's been on our sort of to-do list, but you know, it's only so many constructs you can test. I think what we'd like to do is move away from piggyback and develop feisty 31 based recombination systems where we can test constructs at the same location. Because the system is, so questions, yes, the system's very sensitive to position effects. And initially, we pick driver lines that have fairly high expression because they give us 100% female lethality. And so we do ignore that. Um, so we don't do the behavior tests on that many in the end. We've selected the ones that give us the lethality we want, and then do the behavior tests on maybe a couple of them.
Right, that makes sense. Another question. I'm very curious about the failure of RNAi. Could you
Mm.
please expand on the potential reasons for this? Thank you.
Yes. So in Lucilia, uh, the only way we could get complete knockdown and transformation was to use a driver line that was actually active in the ovaries and in the embryos. Now, this DR3 line, which I didn't really go into it much. When we used that, then we got knockdown in the mother. So we think we're reducing the amount of the female form of TRA that's deposited in the egg. And then when we combine that with zygotic knockout, then we got full transformation. Um, but in doing that, most of the XX flies died, and they died because We think because we found showed that the X chromosome expression of the X chromosome genes had gone up. So we think in Lacilia, like in Drosophila, X chromosome dosage compensation works by doubling the expression of X-linked genes in males, and this doubling is inhibited in females to give you equal expression. And we think transformer has a role somehow in inhibiting that doubling in females. So without transformer, we get hyperactivation of the X and
Mm.
lethality. Why we thought the CRISPR approach would work is we made a knockout of transformer where we inserted a DS-RED gene into TRA. And when we make homozygotes, they're fully transformed to XX males and viable. In Screwworm, Alex tried the RNAi approach, and we only made one construct, but that construct caused us a lot of problems. We seem to have off-target lethality going on. Uh, so it didn't work well at all. It must have been something in the construct design.
Mm. Let's see. The next question is about field trial experience. So, can you share more about the Panama field trial experience and what the pathway is to field trials of the newer strains that you've developed or could be developed?
So the Panama experience was these were from one of the early male-only strains. The strain worked quite well under mass rearing conditions. We're able to produce several 100,000 males. They were radiation sterilized, so this was, very low risk field trial. The problem we ran into was in catching the males to monitor the success of the release. We were really trying to get information on dispersal and longevity. The traps that were used then as now use swarm law, which is these mix of really smelly compounds that the females are attracted to. And the traps do catch males, not as efficiently. And its thinking is that they're a bycatch, that they are chasing the females and sort of run into the traps. But by doing male-only releases, there was no females around to chase. So, so the traps were not very effective at catching males. So we had some data that the dispersal was not, it was okay, but not as good as the Jamaica 6. So that was the data we had. But it was really low numbers of males caught in the traps. Going forward, I'm not really involved in this. Alex keeps me, somewhat, informed. So I'm great of it. He's leading all this. He's leading the EPA registration and the back crossing into Pansia and the testing of the fitness of the males after exposure to radiation and trying the lower doses of radiation, which the old data, from back 60s and 70s by sort of Crystal and others suggests that quite a low, maybe even half of dose of radiation should be sufficient to sterilize the males.
Yeah, that's I guess one of the things, and you hit on it, but I did have a question about optimizing the radiation and how that might impact competitiveness. But I imagine if they're getting hit with a lower dose, then that may raise the competitiveness a little bit of those males.
Yeah, we're hoping. Yeah. But otherwise, yes, going through the registration and so now it's open for public comment.
Mm. Um, Here's another question. Any progress in finding the M factor?
Alexis worked on this a lot in Lacilia. And before her, Megan, we had RNA-seq data from early embryos, from single embryos, and we had a lot of candidates. We had a lot of genes on the Y that were expressed in the embryo, but many more than I expected. Early in the embryo, when M needs to be active. She did CRISPR knockouts on some of the candidates. I don't think, if I remember correctly, there was no single gene that when she used CRISPR produced a phenotype. But when she used a mix of guide RNAs, two guide RNAs targeting different genes, she did get evidence that there was having an effect by changing of the splicing pattern of transformer. I don't know whether we'll publish this. The problem was that once we now working with Paul Hickton, we have a pretty good assembly for the Y chromosome in Lacilia caprina. It's a big chromosome full of repeats. And we find that these male determining candidates, there are many copies of them. And so what we thought was, based on the initial assembly, we're knocking out, just a couple of genes, we're knocking out potentially many sites on the Y. So I don't know. We did see the, transformer splicing shift, but I don't know whether that was just because we were essentially shredding the Y. We do have an assembly of the Cochlear-Meyer Y chromosome. It looks much less complex. Assuming we've got most of it, it's a small Y. I think so. We'll probably switch from Lacilia and see if we can figure out what the M gene is in Cochlear-Meyer.
And identifying that factor could go a long way in developing gene drives. So that would be really cool. But
Right.
yeah.
And also,
Mhm.
I didn't put it in the talk, but that's another way to do conditional male only strain is if you switch on the expression of the male determining factor in female embryos.
See, we had another question in about gene drives. They seem to work best in anopheles and rather poorly in many other species, potentially due to differences in DNA repair mechanisms.
Right.
Your thoughts or perhaps hopes on what might happen with screwworms or other flesh-eating flies?
Yes, so we don't know what the homing rate is going to be in Screwworm yet. I haven't assembled a drive. I hope it's going to be good. I sort of shared, for many years, I sort of shared the same concern because we were working on getting a homing drive working well in spotted wing Drosophila. And some of that we've published, and we have some more papers we're writing up now. And we struggled with drive efficiencies that were kind of low, maybe 60, 70% inheritance. But with some modifications, we're routinely getting drive efficiencies now in the high 90s. And hopefully we'll get some of that published. So I'm more hopeful that we can build on that work we've done in spotted wing Drosophila and get an efficient drive working in Screwworm.
Yeah, there can be. This is a little bit my background. I always thought, well, hypothesis that drives didn't work very well in 80s because they're just so replete with repetitive elements that is a strong selection for homologous recombination would just result in everything shifting everywhere all the time. But I guess we'll see. Um, so let's see. I have another question, which I think we've seen kind of a theme across several of these webinars, which is right, the genetic diversity of New World screwworm in South America in particular is not super well assessed. Do you have any comments or concerns about how the genetic diversity in South America might impact the establishment of some of these lines, especially when you think about like CRISPR-Cas9, right? If you have a few nucleotides shifted in your sgRNA, that can really cause some issues. It's a resistance question.
Yes, so it would be great if we had some whole genome sequencing. It doesn't have to be high coverage from flies caught around South America. I think Mackenzie Titian, she's now down in Panama at the ARS lab. She's doing some of this. Hopefully we can do some more and find out what the diversity is. So far from what I've looked at, the guide RNA for double sex is conserved. But of course, the other way that drive efficiency can be impacted is by heterozygosity in the homology arms. Can reduce drive efficiency. So yeah, I think it's at least if we can get more genome sequencing, we should get an idea of what level of heterozygosity we're dealing with.
And then let me ask one last question before we wrap up. I mean, in your presentation, it was obvious, right? You're working with a completely different species in the U.S. because you're not able to work with NWS in the U.S. and then you have to take that work down and replicate it in Panama. And you
Mhm.
mentioned, and we've heard in previous webinars that that policy might be changing due to the kind of the emergency nature of the situation now. But is there anything else working, whether it's a policy or, the ability to, well, I think ability to get the strain is a huge one. Is there anything else that's kind of acts as a barrier or a challenge when working with New World Screwworm that, if it changed, might accelerate some of the research into these new promising technologies?
Well, I mean, that's the big one. And we've applied for a permit to work with New World Screwworm at NC State. Um, and we're trying to figure out just what level containment lab we need to do that. Um, yeah, that's really, I mean, otherwise, we know how to work with this fly from work in Panama. I think, the engineering is doable. It's not easy. It's not Drosophila by any means, but it's doable. Um, I think one of the frustrations I've had over the years is with the NIFA Foundation grants. They're well set up to get research to do basic research on plant pests, but I found it really, I've had no success trying to do basic research on blowflies through NIFA. And that was frustrating not to be able to do some basic research on screwworm that I think would have actually helped in the end with the applied work. You would see that we did get a basic grant from NSF, was the consortium to work on genes that might be involved in the evolution of parasitism.
Yeah, that's a really good point. As a former APHIS employee, there can be some bias there towards plants for sure.
Uh,
Um, and hopefully, well, I mean, maybe the silver lining of this kind of New World Screwworm emergency we're facing I mean, we've already seen some funding opportunities emerge, and hopefully that will continue and diversify so that a lot of this kind of basic research can continue. But all right, I think that'll end us for the questions. Let me go ahead and
okay.
share my screen and I will wrap us up. Thank you again, Dr. Scott, for an excellent presentation. And thank you to all of you for joining us today and contributing to the discussion. This marks the final webinar in our series, The New World Screwworm, Past Lessons, Present Programs, Future Frontiers. Over the past several weeks, we have explored the history of screwworm eradication, current preparedness and surveillance efforts, and emerging scientific and policy approaches that may shape the future of control and potential eradication. Before we conclude, if you have any ideas for topics you would like to see covered in future webinar series related to genetic biocontrol, we would welcome your input. Please feel free to reach out to me directly at jovercash@fnih.org or to my colleague, Brenda Doss at bdoss@fnih.org. We may also be sharing brief polls on LinkedIn over the couple weeks to gather feedback on particular topic ideas, so please feel free to keep an eye out and consider contributing your thoughts. We also encourage you to revisit previous sessions in this series if you happen to miss them, which will be available through the Gene Convene YouTube channel, and to subscribe to the Gene Convene Virtual Institute newsletter to stay up to date on the most recent advancements in genetic biocontrol. Thank you again for your time and participation. Hope you have a great day.